Well, I would first like to thank everyone for coming and taking the time out of your day to listen to my defense. So I conducted a study on the stable isotopic study on the NAMMA hydrology in the Colorado Rocky Mountains. So first, I have a little overview of how my talk will progress on the right-hand side here, and I will start off with an introduction of NAMMAs. So they are potholes that occur in solid bedrock, and they periodically fill with rainwater to form ephemeral pool environments. And they mostly occur in arid and semi-arid environments, and they generally form in granite and sandstone, though they can form in a whole variety of rock types. And the morphology of NAMMAs is broad. They can be anywhere from a couple centimeters wide to millimeters deep, to up to several meters wide and many meters deep. Each NAMMA acts as its own ecosystem, which gives it the potential to be developed as a model system to explore ecological and evolutionary experiments in the field. So when NAMMAs periodically and temporarily fill with rainwater, they form ephemeral aquatic habitats. And these habitats have specific seasonal environmental conditions that are composed of multiple short and usually unpredictable hydroperiods. And a hydroperiod is depicted in this image here, where it's the time from the initial water input from rain until the NAMMA completely dries up. And so these hydrologic conditions are highly variable, and they have both spatial and temporal variation. And the main determinants of biology in temporary waters have been found in previous study to be the duration of time the pools contain water, which relates to its hydroperiod, and the salinity, so if the waters are more fresh or saline. There's a high selective pressure on biota in these extreme conditions, and that can influence the life history adaptations that they have. And a previous study found that development rates and species richness depend mostly on the residence time of the water and the rate of water loss, as opposed to the actual volume of water in the pool. And this image down here shows some of the diversity of life that can live in these pools. So anywhere from fairy shrimp and tardigrades, and a whole diversity of bacteria and microbes that can also live in the system. And this image on the right shows a desiccated microbial mat that is another example of organisms that survive in these environments. So the major source of environmental variation is the wet-dry cycles. So from water accumulation due to rain and to water loss, there are two very extreme forms of these NAMMAs. And so there's either a time where water is very abundant, and then long periods of time where there is no water at all and it's completely desiccated. And so these hydrology extremes may be the dominant selective pressure of NAMMA biota. And to further explore this, the processes of water loss should be constrained in NAMMAs. And previous studies of different NAMMAs and NAMMA ecosystems consider evaporation as the sole form of water loss. And the goal of my study was to evaluate this water balance assumption. So for this experiment, I considered three ways through which NAMMAs may lose water. The first is evaporation, which is the currently accepted model. The second is seepage through the rock substrate. And the third is a combination of the two processes. So in order to trace the movement of water throughout the systems, I conducted a stable isotopic study. And next, I will go over a little bit of a background of stable isotopes. So in the natural environment, there are two isotopes of water, hydrogen 1 and hydrogen 2. And there are also three different isotopes of oxygen, 16, 17, and 18. And I'll just use hydrogen as an example here. So hydrogen 2 is the heavy species, and it's much, much less abundant in nature than hydrogen 1. And so, since there's such a discrepancy in abundance, the ratio of the heavy species to the light species can be used to track changes in isotopic composition. So stable isotopic data is represented as a delta value, which takes the ratio of the heavy to light isotope in your sample and normalizes it with a standard. So the standard I used was the Vienna standard of mean ocean water in order to compare data across different data sets. And just as a note, delta values are expressed in per mil. So the properties of stable isotopes can be used to track the movement of water through the hydrological cycle. And so down here, there are three most abundant natural forms of water, and ocean water is considered like the zero delta value. So that's the relative standard that we're comparing it against. And so with evaporation, light isotopes evaporate into the vapor phase first, which leaves the remaining water enriched in the heavy isotope. So these lighter isotopes that contain the hydrogen 1 and the oxygen 16 will evaporate and leave a larger amount of the heavy isotopes in the ocean water. And then as these waters move towards the poles and evaporate further, and then also precipitate, the heavier isotopes that are still there precipitate out either in forms of rain or snow. And this trend for the globe can be visualized as the meteoric waterline. And so this plot shows the delta deuterium, which is the ratio of the heavy hydrogen isotope deuterium to the light, and then the delta oxygen 18, which is oxygen 18 to oxygen 16. And these are plotted for all natural surface waters on Earth. So all natural surface waters plot along this linear pattern, and this can be used to as a reference to see how water isotopes should be related before any processes occur. So this gives you kind of the initial starting point of waters before anything happens to them. So this will be applied in my model since we're testing the assumption that water loss in GNAMMAS is evaporation because it's a closed basin. And so when water evaporates, it fractionates, and these light isotopes go into the vapor phase, leaving these heavy ones behind. And so when data from these water samples are plotted, they will be plotted with the meteoric waterline as a reference, and evaporation trends always plot below the meteoric waterline because they start here at any starting point on the line, and then they get heavier and heavier and heavier as those light isotopes leave. So this evaporation trend will be used to see if my data do indeed follow that evaporation trend or not. So a little bit about my methods here. I conducted my study northwest of Lyons, close to Rocky Mountain National Park, and it is a homogeneous slab of silver plume granite surrounded by ponderosa pine forest, and it is a pretty typical habitat for GNAMMAS in the Rocky Mountain region. So my study sites were up in this area. I had 6 GNAMMAS, and then down here I had the remainder of my GNAMMAS. The whole landscape is covered with all different sizes and shapes of GNAMMAS. I just chose these because they were in relatively the same area to make sure the environmental conditions were as constrained as they could be. So I conducted 2 experiments that were approximately 1 week each. The first of which I conducted in August, and I looked at the isotopic composition of 10 GNAMMAS over time. And the water was supplied by natural rain events, and more rain events occurred throughout that week as well. So there was some variation due to the input of rain. And then in November, I conducted another experiment with the same 10 GNAMMAS, but I also had 4 controls, positive controls for pure evaporation processes, such as this one. So I confined water loss to only being able to evaporate out by making the basin a fully closed system by covering the bottom with this thick plastic layer that didn't allow water to leak through. And here's some of my experimental ones that don't have any alterations to them. And there's a variety of morphology and size you can see here. And this experimental, the control is approximately the same size as GNAMMA D here to give you a reference for size. So from each of these GNAMMAs, I took the initial volume of each GNAMMA and then took 2 milliliter water samples throughout the course of time, and then also measured the final volume at the end of my sampling period. So these initial data I plotted on this meteoric waterline that you saw before. So it's the delta deuterium versus the delta O18, and here's the meteoric waterline. And if you recall back to that evaporation trend, both of these data sets from my November experiment and my August experiment show these indicative evaporation trends. So this tells us that evaporation is indeed occurring in these systems, and it is only different between the two because this experiment was filled with rainwater and this one was filled with the well water. So that's the only reason it's offset, is because they have different starting compositions, but the general trend is similar. And another thing to note about this is with the experiment that had the rain events, there is more variability and a greater spread in the data, but that is just due to the inherent isotopic variability that's present in rainwater. And the trends are still parallel. So we can use this experiment with the controls to evaluate the system because the same processes are occurring. So this brings me to my, that last data only shows evidence for evaporation trends. It doesn't really tell us anything about seepage. So in order to evaluate the role of seepage in the system, I developed this hydrology model to apply to my data. So basically I set up that the non-isotopic balance is the change in isotopic composition of water depends on the amount of evaporation and its fractionation, and then the amount of seepage that occurs. And seepage doesn't fractionate. So the water that is present in the NAMMA down here, the composition of that with the isotopes does not change as water seeps. So that stays constant. So my assumptions for this model is that the only two processes that could be at work were evaporation or seepage, that there are no other water loss mechanisms going on. My other assumption was that seepage doesn't fractionate in isotopes, so you won't see any change in isotopic composition if seepage is at play. And also that there's no water input into the system, so that as time goes on, water levels are decreasing. So this model can be depicted in this plot, where you have the natural log of F, which is the fraction of the water left in the NAMMA, on this axis, and so the natural log of 1 is 0, so this point is the initial point where 100% of the water is left in the NAMMA, and as you go along this axis to the left, water is decreasing. And then on this axis, there is the delta deuterium level, so the ratio of the heavy to light. And since I will be plotting the November data series, all of the NAMMAs were filled with the same water from the same well, so the starting isotopic composition is the same for all of them. So all the initial data points will plot down here. And this model constrains my data by two boundary lines, the first of which is pure evaporation processes up here, which is what I constructed those controls to show. And that is where the evaporation to seepage ratio is 1, only evaporation is occurring. The other bound is a pure seepage process where the only water being lost is through the rock substrate, and so that will present a flat slope because there's no change in isotopic composition, it's staying stable. And then these intermediate reference lines show varying ratios of evaporation to seepage, and so the one up here that's 0.30 indicates that 30% of the water loss is due to seepage, and then it decreases down to the 8% of water loss due to evaporation. Sorry. So 30% due to evaporation, 8% due to evaporation, and then the other percent due to seepage. So first I will plot my pure evaporation controls. So these are the ones that had the plastic bottom so that they could only lose water through evaporation, and they do plot on this reference line. So that shows that they were sufficient positive controls to be compared with the rest of my data. And then I will plot my experimental NAMMAs, which their size relates to the size of the NAMMA and the volume of water it could hold. And so these data obviously show a different pattern compared to that of the controls. And this indicates that some other process must account for water loss since they don't plot on this pure evaporation line. So this shows that NAMMAs lose water predominantly through seepage. Anywhere from 70 to 90% of the water is lost through seepage, which is pretty substantial and different from the conventional understanding of water loss at the time. So from this, you can see there might be some relationship between NAMMA size and hydrology, but that would have to be further explored with a larger data set to see if there is actually a relationship through that. But I did develop a potential conceptual model that if smaller NAMMAs lose water predominantly through evaporation and larger NAMMAs lose more through seepage, could be applied to relate NAMMA size and hydrology. So with this diagram, time initial is at the top and then time progresses as you go down the screen. And evaporation is indicated by the E, and Q indicates seepage processes. So starting out with the initial depression of the NAMMA, which can be caused through mineral weathering by lichens or just abiotic weathering by rain. It fills with water, and at first it's assumed that it is an impervious rock basin, and water can only be lost through evaporation. And as evaporation occurs, solutes are left behind in the remaining water, and there becomes an increased concentration in solutes, and so it's more saline. And this salinity can result in salt weathering because these dissolved solids can accelerate mineral dissolution and create a permeable layer in the rock surface. And this can result over time in the formation of these microfractures and the formation of pebbles and rocks in the bottom from the weathering processes. So as this goes on, and as more salt weathering occurs, the NAMMAS come to a stage in development where evaporation is approximately equal to seepage. And this is considered like a mid-developed NAMMA. And then, as time occurs more and more salt weathering happens, the fractures become larger, and then most of the water can be allowed to exit through the bottom of the NAMMA. And this stage is where the predominant method of seepage, or of water loss, is seepage, which kind of was indicated in the previous slide with my data, losing 70 to 90% of water through seepage. And so this model provides a predictive framework that relates NAMMA hydrology and NAMMA size that can be applied to future studies. And applications of this model could alleviate potential confounding of size effects and salinity in NAMMAs by accounting for differential processes of water loss at different stages of development. So here are two examples of my NAMMAs that have this rock debris in the bottom, these gravels, so that could be indicative of the salt weathering that I was discussing. So for some further studies that I would love to look into in the system would be to compare across different climates. So if there's any difference in the hydrology between semi-arid and arid environments or montane and desert environments. I would also like to look at differences in substrate. So if granite and sandstone react differently, or if different granites have different hydrological processes. And one that I would especially like to look at would be seasonality, and look at winter versus summer proportions of evaporation to seepage. As you would expect, in the winter it would be a wetter and colder climate, so less evaporation would occur. And then maybe it's in the winter months NAMMAs lose water predominantly through seepage, and then in the summer, when it's hotter and drier, evaporation will occur at a faster rate, more water will be lost that way, and that will change the hydrology of the system. There are also various biological implications that this model could be applied to. So as I mentioned, lichen can be the initial factors of the weathering and the rock substrate that causes the initial depression. And so I would like to look at if there's any biogeochemical feedbacks between maybe NAMMA distribution or formation or certain characteristics and lichen colonization, and if there's some sort of relationship there. And there's also the potential that microbial mats, like I showed in the beginning, might aid in maintaining water in potholes, so that when they are wet, these microbial mats may seal the mineral grain and not allow as much water to percolate through the base of the NAMMA. So the role of microbial mats in granite potholes could be further studied. So overall, this study constrained controls of NAMMA systems relatively well and allows for this model to be applied to various biogeochemical processes. And this model system could aid in further understanding ecological and evolutionary processes in ephemeral rock pools and the potential selective pressures that may be exerted on the biota in these systems. So NAMMA systems can provide a valuable and controllable framework for investigating environmental processes in vivo. Thank you.
Super, very nice. Cool. The support group, do you have a last word, a quick question or something before I ask you to leave us to the rest of it? Don't have to, yeah, that's fine.
Phenomenal job.
All right. Okay, well, thank you for being here. Thank you for support.
Yes. Let me say hi real quick.
Yeah, hey.
Nice to meet you.
I heard your talk about white.